Hello, my dear students. Hope you having best time and hope you study your lessons hard. In the last session, we studied the types of movement in plants, except the last type, which is continuous movement, such as is cytoplasmic streaming in the plant. We will study today this type of the movement in plant, and we will study the movement in human, okay? We will study the movement in human as example for mammals, okay? Let's see the movement in the plant, which is speak about the cytoplasmic streaming in plant. Movement in plants, continuous movement in plant. This video shows the movement of chloroplast throughout the cell. It is the cytoplasmic streaming or the continuous movement inside the cell or continuous rotation inside the cell, all of them explained when we see this video in Elodia leaf plant. It is aquatic plant. The inner wall of the cell, as you see here, the inner wall of the cell is lined by a thin layer of cytoplasm. The cytoplasm is streaming in a continuous rotation, as you see here. Move throughout the cell in one direction. This movement can be seen by the movement of the chloroplast. This movement helps to rotate the components, the organelles, the nutrients inside the cell. Okay, now we will study the movement in human. The movement of human is example for the movement of mammals. The movement in human controlled by three systems: skeletal system, muscular system, nervous system. Skeletal system, we studied it in the first lesson. Nervous system, you studied it in the last year. Today we will study the muscular system. Okay, let's see. Movement in man depends on three systems, as I said: skeletal system, nervous system, muscular system. Muscular system consists of different types of muscular tissue: epithelial, skeletal tissue, cardiac tissue, smooth tissue. Okay, muscular system consists of three types of muscular tissue. Skeletal tissue, which we will speak about it today, it is striated, voluntary, cylindrical, unbranched, multinucleated, like this, attached to the bones. Okay. Cardiac tissue, cardiac muscular tissue, it is related to the heart. It is striated, involuntary. Okay. Smooth muscular tissue, it is related to the tissue of veins, arteries, blood vessels, stomach, whatever. Okay. It is not striated, unstriated, involuntary also. Muscular system consists of muscular tissue, and it is responsible for movement of the body parts through contraction and relaxation of muscles. Muscles have the ability to contract and relax, and we will study today how the muscle can contract or relax. Okay. It consists of group of structural units called muscles, which also consists of group of tissue called flesh. Muscular system represented by two systems, as I said, voluntary and involuntary. Voluntary, skeletal or striated muscles, they are under the control of human will, as skeletal muscles, which are striated, and stronger muscles. It's represented by most of body muscles. Involuntary muscles, they are not under the control of human will. I cannot control them, okay? As smooth muscles, which unstriated, such as the muscles of blood vessels. Cardiac muscles also striated muscles, such as the muscles of the heart. Cardiac muscles, only the muscles of the heart, okay? Muscular system consists of muscles, okay? And we will study the structure of skeletal muscle. Skeletal muscle consists of group of thin, like this one. This is the muscle, okay? This one is long, thin filaments called muscle cells or muscle fibers. Each muscle fiber doesn't exist alone. What's meaning of doesn't exist alone? Meaning that they always found in groups known as muscle bundle. Each muscle bundle surrounded by a bundle membrane called bremasium, okay? Each muscle fiber or muscle cell consists of protoplasm, this is the living material, which include cytoplasm, this one, look here. This is the muscle fiber. Muscle fiber consists of protoplasm, which consists of sarcoplasm and multinuclei, okay? Sarcoplasm, it is the cytoplasm, okay? So the cytoplasm in the muscle fiber change its name and be sarcoplasm. The cell membrane that surrounds the muscle fiber is called sarcolemma. A group of myofibril, this one. So the muscle fiber consists of group of myofibril. The group of myofibrils, their number ranges between 1,000 to 2,000 myofibrils, arranged longitudinally and parallel to the longitudinal axis of the muscle, okay? So each myofibril consists of group of light discs denoted by I band, symbolized by I, this one, which composed of actin protein, thin protein filaments called actin, bisected by Z line. So again, each myofibril consists of two groups. The first one is group of light discs. This is light discs, which contain, this is the light discs, which contain of thin long filaments, thin protein filaments called actin filaments, bisected by Z discs or Z line. Okay, this is the group of dark filaments denoted by A, this one. A band consists of two types of protein, thin protein filaments, this is the thin protein filament actin, and this one is thick protein called myosin. So the thin protein filament actin form the light discs, while two types of protein, actin or myosin, form dark discs. And this dark discs denoted by A, and this is between them inside the A band, this is a region called H zone, which formed of myosin only. So we have three types of area. Group of light discs formed from actin protein, this is the thin proteins, and group of dark discs which contain two types of proteins, actin and myosin, the thin actin and thick myosin, and area of semi-light band which consists of myosin filaments only, denoted by Z zone. So we have I zone, we have A band or I band or I zone, A band, and H zone, which consists of myosin filaments only. The sarcomere is the distance between two successive, this is the Z line and this one is the Z line, okay? Z line, the distance between Z line and the successive Z line, the successive meaning the following one. So the distance between two successive Z line is called sarcomere, which found in the middle of light band, Z line found in the middle of light band. This blue line represents the protein, thin protein filaments, which is actin, and the Z line present in the middle of the protein actin, okay? We can summarize what we said, that each myofibril consists of light bands I, semi-light bands H, dark bands A. Light bands consist of actin filaments, which is thin filaments, and the actin filament mediated by dark line Z. Semi-light bands H consist of thick filaments called myosin only. Dark bands A consist of two types of protein. The thin one is actin, the thick one is myosin. Okay? This is the consists of each myofibril, the components of each myofibril. Okay. Also, we need to remember or to study and keep that in one myofibril, the number of semi-light bands H equal the number of dark bands A equal the number of sarcomeres, all of them equal to each other. The number of Z lines equal the number of sarcomeres plus 1. If we would like to calculate the number of Z line, we will see the number of semi-light bands or the number of dark bands A or the number of sarcomeres, and we will add number 1. Okay? Here you don't need to keep number of sarcomeres only, number of sarcomeres or number of semi-light bands or number of dark bands, and add number 1. Okay. Before studying the muscle contraction, let's study that the sarcomere is the distance between two successive Z lines that are found in the middle light bands in the myofibril. Also, the least number of myofibrils in the skeletal muscle equal the number of muscle fiber multiply 1000. Because we said that the myofibril ranges from 1000 up to 2000. So the least number is the number of muscle fiber multiply 1000. And the largest number or greatest number of myofibrils will be the number of muscle fiber multiply 2000. Okay? How the muscle contract? The muscle contraction, the muscle have the ability to contract and relax, as we said before. Any muscle in our bodies have the ability to contract and relax. Skeletal muscle, which is striated, voluntary, cylindrical, unbranched, multinucleated, have the ability to contract and relax also. And this is due to coordination among skeletal, nervous, and muscular system. Okay, let's see how the transmission of nerve impulse into skeletal muscle. How is the nerve impulse transmit from nervous system to skeletal system or skeletal muscle to skeletal muscle? Okay, you studied it in the last year. There are 3 states of the cell. Rest state, which is polarization state, and this is before the voluntary skeletal muscle received the nerve impulse, where the outer surface of the muscle fiber carry the positive charge and the inner surface carry the negative charge. Potential difference is arising between the outer and the inner surface, called polarization state, cause what we said polarization state. So the polarization state is the state of muscle fiber membrane when the outer is positively charged and the inner is negatively charged. The second state is depolarization state. This state arises when the voluntary skeletal muscle receives the nerve impulse. The stimulus caused the skeletal muscle contraction is considered the arrival of nerve impulse from the brain and spinal cord through the motor nerve cells, whose endings are firmly connected with the muscle fiber, forming synapse neuromuscular junction. You study it. Okay. When the nerve impulse reaches the synaptic vesicles, we will explain it, cause the alter the charges between inner and outer surface. The rest state followed by depolarization state, or we can say that the polarization state followed by depolarization state, then we would like to return back to rest by a state called repolarization state. Okay? This is the 3 states. The first one here, it is the rest state or polarization state. The outer is positively charged and the inner is negatively charged. The potential difference between them called polarization states. And this one is depolarization. The voluntary skeletal muscle receives the nerve impulse. When the nerve impulse reaches the synaptic vesicles that are present in the endings of the nerve cell, the calcium channels, and you will see the picture, explain this later. Calcium channel in the cell membrane passes the calcium ion inside the nerve cell, causing the rupture of synaptic vesicles and to release some of neurochemical neurotransmitters called acetylcholine. The neurotransmitter crosses the synaptic vesicles, causing alternate in the charge. The outer will be negative and the inner will be positive. This depolarization will transmit along the muscle. Okay, this one repolarized, while the neighboring one is depolarized again. We see here the stimulation state depolarization. That's what I said now. When the nerve impulse reaches the synaptic vesicle that present in the nerve cell. The calcium channel, this is the calcium channel in the cell membrane, passes the calcium ion inside the nerve cell, causing the rupture of synaptic vesicles. This is synaptic vesicles which consist of chemical neurotransmitters such as acetylcholine. The neurotransmitter cross, as you see here, cross the synaptic cleft that is present between nerve endings and the membrane of the muscle fiber till reaching the surface of voluntary muscle fiber. The potential difference of the two sides, this is the two sides of the muscle fiber membrane, change. The outer will be negative and the inner will be positive. And this is due to the increase of the cell membrane permeability. This alternation of charges is due to increase the permeability of positive ion sodium, which pass through the muscle fiber membrane rapidly from outside to inside. This is the sodium ion passes from outside to inside, okay, causing the depolarization of the muscle fiber membrane and leading to muscle contraction. So this is the depolarization state, which we can define as it is the state of muscle fiber membrane where the outer surface is negatively charged and the inner surface is positively charged. Repolarization, or returning to repolarization to rest state. This is called repolarization states. After a part of second, the potential difference of the muscle fiber membrane return to its normal, which called repolarization, return to rest state. Due to action of cholinesterase enzyme, this is the cholinesterase enzyme which bind with acetylcholine to break down it, and cholinesterase enzyme found abundantly at the neuromuscular junction, this area, neuromuscular junction, this area, and work on destroying the acetylcholine substance and convert it into choline and acetic acid. This is the choline and this one is the acetic acid. So its action stop. When we break down the cholinesterase, when the cholinesterase enzyme break down acetylcholine, its action will stop. So it effect vanish and the muscle fiber membrane permeability to ions return to its original state at this rest state, which is the outer will be positive and the inner will be negative. Okay, this is the rest, returning back again to the rest state by the action of cholinesterase enzyme. Don't forget that. Return to polarization states by the action of cholinesterase enzyme. Okay, let's see the muscular contraction and relaxation according to what you studied this year. Mechanism of muscle contraction according to Huxley's theory on sliding filaments. This is the new part of lesson. Muscular contraction and relaxation according to Huxley theory of sliding filaments. It is the most acceptable theory that explains the muscle contraction on sliding filaments. The idea of sliding filaments that this theory depends on ultra-microscopic structure of muscle fibers, where each muscle fiber consists of group of myofibrils, as we said, and each myofibrils consists of two types of protein filaments. Each myofibrils consists of two types, which, as you see, actin, thin actin, thick myosin, thin actin, thick myosin, thin actin only, light band, actin plus myosin, dark bands, myosin only, semi-light bands. Okay. This is the actin, this is the myosin, this is dark. The area, the sarcomere is from Z to successive Z. Don't forget these names. Okay. The first is the actin and the second is myosin. Huxley used the electron microscope, which has the ability to magnify for about 1 million in comparing to muscle fiber at a state of contraction with another muscle fiber in a state of relaxation. And conclude that at contraction, look here, this is at rest state, this is at contraction. This is, we can see here it is rest state, relaxation or polarization. This is the stimulation, contraction, depolarized, and this one is returning to rest state, meaning repolarization. Okay. As you see here, the length of the myofibril decrease in contraction. The protein filament forming the muscle fiber slide over, as you see, slide over each other, cause contraction or shrinkage of muscle. There is the transverse length. There is transverse length extended from myosin filaments to attach with the actin filaments. Okay, this is the actin filaments and this is the myosin filaments. There is a transverse link from myosin filaments to actin filaments by the help of calcium ions and ATP. We consume ATP while the muscle contracts, we consume ATP, and while the muscle relax also we consume ATP. So we consume ATP during contraction and during relaxation. Here we make contraction, we consume ATP and calcium ions. While relaxation, we also consume ATP and release calcium ions. Okay. The transverse length acts as hooks. As in the presence of stored energy in ATP, the direct ATP, don't forget that the ATP is the direct store of energy in the muscle, which pull, so the hooks of myosin filaments pull the knife-boring grooves of actin filaments. This is, look here, this is the hooks of myosin filaments, pull the protein filaments of actin from both sides toward each other, leading to muscle fiber contraction. During contraction, the Z line become closed to each other, like here. This is the normal state or rest state. This is the contraction. The Z line become close to each other. What is meant by transverse link? They are filaments extended from myosin filaments, like hooks, to reach to actin filaments by the help of calcium ion and ATP. We can summarize what we explained in the following table. Okay. The light band during contraction, its length decreased due to approaching of actin filaments from each other, and also semi-light band its length decrease or disappear according to the strength of muscle contraction. Dark band A, its length doesn't change. Dark band A, which formed from myosin only, its length doesn't change. Sarcomere, the distance from Z line to Z line, its length decrease due to approaching of two Z lines together. Myosin filaments, remember myosin filaments, it is the thick protein from which the transverse links are formed that work as hooks to pull knife-boring grooves of actin filaments by the help of energy stored in ATP and also calcium ions. Don't forget both ATP and calcium ions. Okay. Actin filaments, they approach each other in the direction of semi-light bands. They move toward the semi-light bands, H, so the light band decrease. Z line, they approach each other, so the length of sarcomere decrease. Okay. At the relaxation state, when the stimulus disappears, what happened to the muscle cell? The transverse links will move away from each other, move away from actin filaments. Transverse links, which arise from myosin filaments, move away and leave the actin filaments. Therefore, the muscle relax, also by consuming a part of energy stored in ATP molecule to separate the transverse links from the actin filaments. So, we consume ATP during contraction of muscle or during relaxation of muscle. But we consume calcium ions only during contraction. The Z line move away from each other, therefore the sarcomere return to their original length. So don't forget that during contraction we consume ATP molecules and calcium ions. During relaxation we consume also ATP molecules. If the amount of ATP doesn't enough, so the muscle may contract only, can't make relaxation, and this is called muscle fatigue, or the muscle cannot contract at all. Okay. From the previous, we can conclude that the attachment of transverse links with actin filaments during the muscle contraction and the separation of transverse link from actin filament during the muscle relaxation need ATP molecules, so that as I said, we consume ATP during contraction and relaxation, but we need calcium ions during contraction only. According to the sliding filaments theory, we can make the following comparison. The first one, the muscle fiber at contraction states the transverse links are extended from the myosin filaments, reach to actin filaments, then pull them toward each other. Therefore, the muscle contract and the sarcomere length decrease. The Z line approach each other, so the length of sarcomere decrease. It needs acetylcholine and calcium ion and the energy stored in ATP molecules. Acetylcholine to transfer the nerve impulse from the nerve cell to the muscle cell, okay? The muscle fiber at relaxation don't need all of them. The transverse link move away from the actin filaments, leave the actin filaments by the help of ATP, energy stored in ATP molecules. Okay, therefore the actin filaments separate from the myosin filaments and the muscle relaxation relax, and the Z line move away from each other. Therefore, the sarcomere return to its original length. Now we will study what happens if the muscle perform a vigorous effort. We will have muscle fatigue. What caused muscle fatigue? The causes of muscle fatigue may be perform a vigorous effort, suddenly and vigorous motion leads to rapid and successive muscle contractions, which cause shortage of oxygen in the muscle. So the muscle need to make anaerobic respiration of muscles, which cause or leads to accumulate of lactic acids and also decrease in ATP molecules. Accumulate of lactic acids leads to muscle fatigue, while decrease in ATP molecules leads to painful muscle spasm. Now we finished studying the first chapter which speaks about the support and movement in human and living organisms. Best wishes for you.